Hello and welcome to the Genetics Lab Practice Dilution Problems Solutions video. This is a video working out the sample dilution problems. These same solutions are available in text form on Blackboard. If you're still having problems after watching this video, I strongly urge you to see your TA or me or another instructor and get some help. Dilution problems are tricky for a lot of people, a lot of students, a lot of people off the street, but once you get past a couple hurdles, they become very easy. So it's worth getting a little help, and then you'll find that they stop bothering you and they become really a joy for the rest of your life. So let's just jump right into it. We start off with probably some easy things for you, which is just identifying simple dilutions. And of course, simple dilutions aren't too bad to figure out, right? And that is identifying what dilutions are you making. And so what dilution are you making if you add 0.1 mils to 0.5 mils? That's 1A. And of course, the way you figure out what dilution you're doing is you take the sample, 0.1 mils, and you divide it by the sample plus the diluent. And that's important. It's not the sample divided by the diluent, so this is not a 1 to 5 dilution. It's the sample divided by the sample plus the diluent. So this is 0.1 divided by 0.1 plus 0.5. We can solve that by adding these two together. So that's 0.1 mils divided by 0.6 mils. It's very messy to have 0.1 mils and 0.6 mils. We can get rid of those decimal points. So this is a 1 mil over 6 mil dilution. In fact, notice that there are units on the top and the bottom that are the same. So we can divide them out. We can cancel the units. This is, in fact, a 1 to 6 dilution. And you'll see that written in a number of different ways. 1 over 6, 1 colon 6. It doesn't matter how you write it, 1 dash 6. But it's a 1 to 6 dilution, a 1 to 6 dilution. So that's the way you do a dilution. Very simple. And now that you know how to do it, these should all be pretty simple, right? If we tackle B, it's going to be basically the same thing, right? On the top, we have our sample, 0.1 mils, and I'm going to leave off the units because we know they're going to cancel out in the end, right? So 0.1 mils over 0.1 plus 2.6, that's going to be 0.1 over 2.7, and that's going to solve to 1 to 27 when we get rid of those decimal points, when we move it over. We always want a 1 on the top. That's going to be very important later on, especially when we get to serial dilutions and we want to turn these dilutions to dilution factors. It's going to make the math much, much easier. Always get a 1 on the top. C, here we're using 0.2 mils of our sample, our culture, and we're adding it to 4.8 mils of diluent, of broth. So that gives us 0.2 mils in a total volume of 5 mils. That's a 2 to 50 dilution. We want to make sure that we have a 1 on top. So this is really a 1 to 25 dilution. And again, it's important to have a 1 on top. So very simple, right? Okay, so if this is simple, then let's see if we can do dilutions, if we can design some. And that's what it asks you to do in question 2. Design some dilutions. So for A, it says design a 1 to 2 dilution. And so the temptation here is to say, well, I'll take 1 mil of my sample and add it to 2 mils of diluent. But remember, what we're going to do from the last example was you're going to take your sample, you're going to divide it by your sample plus your diluent. Well, that would be 1 over 1 plus 2. That would be a 1 to 3 dilution, so that isn't going to work. So we have to subtract our sample from our total on the bottom. So if we're going to add 1 on the top and on the bottom, then our total needs to include that 1. So if it's going to be 1 to 2 and there's a 1 on the bottom, that 2 on the bottom is 2 minus 1, so it's 1, right? So if we're going to do this with 1 mil, and we could do it with 1 mil, 1 gallon, 1 pint, 1 liter, 1, you name it, but let's keep this with a normal lab volume, so we'll call it 1 mil. Then this would be 1 mil of sample. That 1 mil of sample needs to be added to the bottom as well. And to make it a 1 to 2 dilution, it would need to be added to another 1 mil, right? Because 1 over 1 plus 1 is a 1 to 2. Now, of course, the units cancel out, and that gives us our 1 to 2 dilution, right? Now we can do that with very clean numbers like 1 over 2, and sometimes it's very easy to do that. We can do 1 mil or 1 gallon or 1 pint. In B, we could do that. For example, if you want to do a 1 to 38 dilution, what could we do? We could add 1 to what? What would you add it to? You can pause here and think about it. In fact, I suggest you pause here and do B and C. Alright, I'm going to assume that you paused and did B and C without looking at the answers from Blackboard. And what you probably did, if you did B and C like A, is you added 1 to 37, right? And 1 to 49, because that's the way A was done. But that isn't the way you have to do it, right? Because we can change it. So for B, for example, we can alter the volumes. Maybe we don't want to deal with 37 mils. Maybe that's too big a volume. Maybe we don't have 37 mils of diluent. Maybe it's precious stuff that we're dealing with. So we could have 0.1 mils of our sample instead of 1 mil of our sample, and add it to 3.7 mils of diluent. So 0.1 mils plus 0.1 mils, 0.1 mils into 3.7 mils. So that 0.1 mils goes on the top and the bottom. 3.7 mils on the bottom. So does this work out? Well, if we solve it, that's 0.1 mils over 3.8 mils. That's a 1 to 38. Mils, of course, cancels out, so yes, it is 1 to 38. So why is this 1 to 38? Well, remember, when you're doing fractions, remember this back from your old math class, when you have a fraction, you can do anything you want to this fraction. Multiply it by 1 or 10 or 0.1 or 0.5 or 432, as long as you do it to the top and the bottom, it doesn't actually affect the number, right? So here we took 1 and 38, multiplied it by 0.1, or divided it by 10, right, to get it to 0.1 and 3.8 to deal with numbers that we found a little more convenient to work with. And it was still a 1 to 38 dilution. So we can do a 1 to 50 dilution. We can do that the same way in smaller dilution. That would be 0.1 and 4.9. Again, a smaller sample to work with, much easier. That would be 0.1 and 5. That's 1 mil and 50 mils, or a 1 to 50 dilution. Is that the only way to do it? Of course not. There's many other ways we could do that. In fact, if you look at question 3, it says come up with at least 2 different ways to do the next 3 dilutions. And I can tell you there are many, many more than 2 different ways to do these dilutions. So I'm going to show you 2 simple ways to do each of these 2 dilutions, but you could come up with many more than these 2 simple ways. So, for example, A. The obvious way, 1 into 99, is a 1 to 100 dilution, right? Well, we can divide that by 10. That's 0.1 into 0.1 plus 9.9. That's 0.1 into 10. That's a 1 to 100 dilution as well. We divided everything on the top and the bottom by 10, right? We divided everything by 10. Just put a decimal point in front of everything. It's still a 1 to 100 dilution. We could have divided everything by 10 or 100 or multiplied everything by 3. It still would have worked. The number that you're going to choose is going to be by what's convenient to work with. We could have broken it up into more than one dilution, but that's something we're going to talk about in a moment. B. The obvious one, of course, 1 into 74. That's 1 into 1 plus 74. That's a 1 to 75 dilution. Well, what else could we do? We could divide it by 2. That would be 0.5 into 0.5 plus 37. Let's see, does that work? That's 0.5 into 37.5. Let's see what happens if we solve that. If we multiply both the top and the bottom by 2, that's a 1 to 75 dilution. Of course it does. Whatever you do to the top and if you do the same thing to the bottom, it's going to work out. I don't know why that would be an easier number to work with in the lab, but if it is, go for it. C. 1 to 88. Well, the obvious way, 1 into 87. So always start with that, and then you can adjust the numbers to fit your lab volumes. Usually anything from 1 microliter to 10 mils is a number that's very convenient in the lab. Maybe 100 mils. Anything outside of that, it starts to become inconvenient. So that gives us a 1 to 88. What else could we do? Well, we could divide that by 5 just to make it really strange. So what would that give us? 0.2 into 17.4. All right. That gives us 0.2 into 17 over 17.6. If we multiply that out, that's 2 over 176 to get rid of those decimal points. If we divide that by 2 to clean it up, that's 1 over 88. So I'm not saying these are the easiest way to do it by any means. In fact, they aren't the ways I would choose to do it, but it works. You can see there are lots of ways you can do it. Dividing by 10 is usually a good way to choose, but not always the easiest. 4 and 5 are certainly the problems that are going to give people some trouble. But if you can do 1, 2, and 3, you'll be able to do 4 and 5. So 4 and 5 are serial dilutions. And serial dilutions require only what you knew for 1, 2, and 3, plus remembering one thing. This formula. Initial concentration equals final concentration plus dilution factor. And remembering, of course, that a dilution factor is 1 over the dilution. Okay, and this is why you want to have your dilutions solved so that you have a 1 over them, because it makes it very, very easy to turn your dilutions into dilution factors. It makes the math so easy you don't need calculators. At least, not unless you have to multiply a whole bunch of them together. So this looks really tough. But what you do is you figure out what's the question and you tackle it. Okay? So, what do we got here? We got a culture flask. We don't know what's in it. Some kind of cells. We don't know the concentration, but we know that it's been diluted through 4 tubes, and then a sample was taken out, put on a plate, and some colonies grew. And so here's the thing. Initial concentration, final concentration, and dilution factor. You are always going to know 2 things if this is going to be solvable, and you do. You know, or can know, the final concentration because we have colonies growing from this tube. That's our final that's where we're going to get our final concentration. And you have the dilutions spelled out to you, and that's going to give us our dilution factor. So this is a problem that's asking you the initial concentration. You're always going to be told 2 of them and get asked for the third. They're always like that. And the thing you have to remember when you look at a dilution problem is that the concentration goes down this way and up that way. It goes down this way and up that way. That's what you have to remember. It's most concentrated in that initial flask, least concentrated after it's been diluted several times, right? Makes sense. If that's not what you get after you do your calculations, then you're just plugging in formulas and not thinking about it. You don't want to do that, right? If the result doesn't make sense, you should go back and check your calculations. So, first question here, A, is what is the total dilution factor? So, to get the total dilution factor, we're going to have to work out all these dilutions and turn them into dilution factors. And you know, that's the best way to figure out the whole problem anyways, is by calculating each dilution, turn it into a dilution factor, and then multiply them together and get the total dilution factor. So let's do that. Tube A, the way tube A was made from the culture flask is 0.1 mils was taken from the culture flask and added to 0.9 mils of broth. So here's tube A, and it was 0.1 mils added to 0.9 mils. Well, we know how to do that. This was like the first question, right? We know how to solve that. That's 0.1 mils over 1 mil. Mils cancels out. So that's a 1 to 10 dilution. So remember, a dilution factor is 1 over the dilution. So 1 over 10 is 10. That's why we want a 1 over the 1 in the dilution. It makes it very easy to solve. 1 over 10 is 10. So the dilution factor is a nice clean number. Okay, tube B is 0.2 mils from tube A added to 0.8 mils of broth. So the way we solve that is 0.2 mils into 0.2 plus 7.8. That solves to 0.2 over 1. We can clean that up into 2 over 10. That's not all the way solved. We want to clean that up a little bit more. That becomes a 1 to 5 dilution. And of course, that becomes a dilution factor of 5. Tube C is 0.3 mils from tube B added to 7.2 mils of broth. So that's 0.3 into 0.3 plus 7.2. So 0.3 mils of sample over 0.3 mils of sample plus 7.2 mils of diluent. So that's 0.3 over a total of 7.5. That becomes 3 over 775. We can solve that to 1 over 25. That's a 1 to 25 dilution, or a dilution factor of 25. And tube D, the final one, is 0.1 mils from tube C added to 0.9 mils of broth, which is the way tube A was made, so we know how that's going to work. That's 0.1 mils added to 0.9 mils, gives us 0.1 in 1, which gives us a 1 to 10 dilution and a dilution factor of 10. All right. So, total dilution factor, how do you get that? You just multiply them all together. And I can tell you, here is one place that a lot of students make a mistake. Multiply all the dilution factors together. Don't multiply all the dilutions together. It isn't that this will give you the wrong answer. It will if you do the math correctly. But a lot of students forget how to correctly multiply fractions and how to do it correctly in a calculator, right? Especially if you don't solve them all the way. But it's very hard to mess up multiplying 10 times 5 times 25 times 10. That's a very clean math problem. So solve them all the way to dilution factors. And if you do that, especially if you use scientific notation, here 10 to the 1, 5 times 10 to the 0, 2.5 times 10 to the 1, 10 to the 1. The math is going to be very easy. You're going to get 1.25 times 10 to the 4. That's your dilution factor. 10 times 5 times 25 times 10. B. What is the concentration of bacteria in the flask? Well, that's another way of asking, what is the initial concentration, right? Because that's how you made tube A. You took a sample from the flask. Well, IC equals FC times DF. So this is DF. We need FC to get IC. So, how do we get FC? Well, FC comes from the sample that was taken from tube D. Tube D is the most dilute tube, the final concentration, and 0.1 mils was taken from it, and 100 colonies grew from that 0.1 mils on a plate. So we know that the final concentration in tube D is 100 colonies, 100 colony forming units per 0.1 mils. Now again, that's an ugly concentration. We don't really want cells or colony forming units per a fraction of a mil, 0.1 mils. We'd really like to have this be a clean concentration. We'd like cells or colony forming units per mil. So since this is per 0.1 mils or per tenths of a mil, how do we solve this to per whole mil? How would you do it? Right, we need to multiply it by 10, or 10 over 10, right? Because if you multiply this by 10, that brings it to a whole mil. As long as we do the same thing to the top and bottom, then it hasn't changed this at all, right? That's what we were just talking about on the last slide. So multiply this by 10 over 10, that's 1, right? And what we'll get is 10 to the third, or 1,000, colony forming units or cells per mil. That's our final concentration. Well, now we have FC and DF. That gives us our IC, which is the concentration of bacteria in the flask. So we can do our calculation. IC equals 10 to the third colony forming units per mil times DF, 1.25 times 10 to the fourth. That doesn't have any units. Multiply it out, you get 1.25 times 10 to the seventh colony forming units per mil. Very easy. C. What is the concentration of bacteria in tube C? This is a question that a lot of students find very tricky. But in fact, it's very easy. All you have to do is redefine the initial concentration. So, if you want to know the concentration of the of the bacteria in here, tube C, then just pretend that the experiment started here in tube C. And say, this is the flask. This is where the experiment started. This is the first tube. You can even draw a little flask right here and say that the very first dilution came from here, the flask, into tube D. And so what was that dilution? It was a 1 to 10 dilution. We know what the final concentration was. It was 10 to the third. We know what the dilution was. It was 1 to 10. IC equals FC times DF. Well, you know those two numbers now. IC equals FC times DF. Concentration in here, 10 to the fourth colony forming units per mil. Very easy. What's the concentration in tube B? IC equals FC times DF. FC is 10 to the third. The DF would be 1 to the 10 times 25. So 250, right? Want tube A? 10 times 25 times 5. Go all the way back here, and it was 10 times 25 times 5 times 10. That's how you got to the initial flask. Move this way, it should go up. You're multiplying all the DFs together. Go this way, and the concentration should go down. And in fact, you can do this either way. You can call this the IC, this the FC, and work from this direction. Oops. Or you could have done it the other way. Since you have now figured out the initial concentration, you could call this the FC and work that way. But that would have been harder since there were more tubes in between. But if you were asking for tube A and you knew the initial concentration, you could have done that if you preferred. Okay? All right. The big one now. You ready? Question 5. A culture flask contains E. coli at a concentration of 4.5 times 10 to the 8th colony forming units per mil. diagram how to, and then you're given 3 different chores. So this is a backwards dilution problem compared to what you've seen before. How to take a concentration of a culture of known concentration and dilute it enough to give you something that's usable. This is something that you might actually have to do in a lab for real, as opposed to the kinds of things that you've been asked on paper. Something I have to do every time I prepare a sample for you. So dilute it this time such that you can obtain 45 colonies on a plate. Something usable, right? Okay. Always start by thinking IC equals FC times DF. I just write it right there on the top of the plate. Excuse me, on the top of the paper. Alright. So what do we have this time? Well, we have IC, 4.5 times 10 to the 8th CFUs per mil. And we know where we want to get 45 colonies on a plate. And that's going to give us the final concentration, right? So we know we're going to have IC and we're going to have FC. So that means in this problem we're working towards the DF. You're always going to know 2 of them. So let's do A. The final concentration here is going to be the concentration of the cells in the last tube. Remember, it's not talking about the concentration on the plate. It's the cells in the tube that are sampled to make that plate. Now you can technically plate whatever you want, but 100 microliters or 0.1 mils is typical, like described in part C. So let's just assume we're going to use 100 microliters. If you change it, you're going to make it more complicated. But let's just keep it at 100 microliters for now. If you want, you can do something else. If we plate 100 microliters, then 45 colony-forming units. Each colony comes from a colony-forming unit, or cell. 45 colony-forming units comes from 100 microliters, or 0.1 mils. So 45 per 0.1 mils gives us a final concentration of 450 colony-forming units, or cells, per mil. So that's our final concentration, 450, or 4.5 times 10 to the 2 in scientific notation. So now we have our IC and we have our FC. How do we get our DF? Well, if IC equals FC times DF, then we can just divide IC by FC to get DF, right? So IC divided by FC equals DF. 4.5 times 10 to the 8th divided by 4.5 times 10 to the 2 gives us our DF. Well, 4.5 solves very nicely. 10 to the 8th divided by 10 to the 2 gives us 10 to the 6. Units cancel out. That gives us 1 times 10 to the 6th. That's our dilution. Well, that's a dilution you've seen before, isn't it? That's one we can do in a lot of different ways, in fact. And hopefully you've seen from the first couple questions that you can do this any way you want to do it. In fact, you could do the one that you saw in the Newcomb experiment or in the streptomycin experiment. You could do 3 1-to-100 dilutions, and in fact that's the easiest one to do. Or you could do 6 1-to-10 dilutions, or some combination there. Perhaps you'd like to do 3 1-to-10 dilutions and a 1-to-1000 dilution, if you have the volume of diluent to do that. Or maybe you'd like to do some weird combination of dilutions, a 1-to-50 and a 1-to-20 and a 1-to-50 and a 1-to-20. It really doesn't matter what you do, as long as when you multiply them all together, they multiply out at a 10 to the 6th. That's all that's important. So the easiest one to do, of course, is 3 1 to 100 dilutions. 10 microliters into 990 is a 1 to 100 dilution. 10 into 990 becomes 10 into 990 plus 10. So 10 into 1000, that's 1 to 100. Do that 3 times, and you've got a 1 to 1 million, or 10 to the 6th dilution. Plate 100 microliters, that's what you were asked to do, and you'll get your colonies. Very simple. B. So what if instead of 45 colonies you want 100 colonies? Well, obviously that is changing the final concentration, right? Didn't change the initial concentration, but it did change the final concentration. That means it's going to change the dilution factor, right? So final concentration is still the concentration of the cells in the last tube. So let's still plate 100 microliters. This time we want 100 colony forming units per 0.1 mils, so that means we have 10 to the third colony forming units per mil. We're going to use the same formula to get our dilution factor. IC over FC equals DF. That gives us 4.5 times 10 to the 8th, that's our IC, divided by our FC, 10 to the third, equals DF. This time we have 4.5 times 10 to the 5th as our dilution factor. And again, you can break this down any way you want. A simple way to break this down would be to pull out 1 to 100 dilutions until you have a simple dilution left. And what that would do would be to give you 10 to the 2 times 10 to the 2 times 45. That's an easy way to do it. And so what that might look like, for example, you know how to do all of these because this was question 3. Might be like this. Here are 2 1 to 100 dilutions. And of course there are lots of ways to do a 1 to 45 dilution, but 10 into 440 microliters is one way you could do it. You certainly could do something else, but that'll work. You can pause this if you need to look at it or work it out. 10 over 440 plus 10 becomes 10 over 450. That's 1 to 45.